It's my very great pleasure to introduce our speaker for today. Dr. Yun is a professor of systems agronomy in the Department of Plant Sciences, where he has a history of researching so many things that I'm not going to list them, but the confluence of beautiful research looking at nutrient management and soil health and performance and innovation for efficiency of fertilization and crop management systems in order to increase yield and understand both theory and practical recommendation for management of agronomical systems. Today we're going to hear a little bit about that. Of course, Dr. Yun is going to tell us about one aspect of his research, but he has a really beautiful, varied research portfolio, has written over 100 refereed journal articles and many more publications related to his research, and I encourage you to check out many of them. I'm not in his field at all, and I had fun reading through some of them. So not only for specialists, but I assume even more fun for specialists. He has an amazing track record of funding and institutional service in a diversity of roles, including being a senior associate editor for Agronomy Journal, and president of the Tennessee Agricultural Production Association. So for all of those reasons and many more, I'm very excited to listen to his talk today and to introduce him to you. Dr. Yun, take it away.
Thank you, Nina. Good afternoon, everyone. Welcome to attending. Today, I'm going to take this opportunity to present some research relating to soil, crop, and environment health. The first project I'm going to talk about is grain health related to heavy metals after long-term poultry litter application. Poultry production is an important industry in Tennessee and the Southeast region, including Georgia, North Carolina, Alabama, Arkansas, and Mississippi. Poultry litter is a cheap and effective source for crop nutrients. Poultry litter application has been a good practice to enrich soil nutrients and improve soil health. Poultry litter has positive effects on soil properties, including increasing soil aggregate stability, available water content, and soil carbon and nitrogen storage. At present, poultry litter is commonly used based on nitrogen requirement. However, it also contains other nutrients such as phosphorus, potassium, magnesium, and many heavy metals. Actually, poultry litter can also be used as a phosphate fertilizer. But because if it is used as a nitrogen fertilizer, the application rate is higher. So more and more producers, they use chicken litter as a nitrogen source. In previous studies conducted in our state, soybean yield, soil carbon storage, and soil PNK contents were significantly increased after long-term applications of poultry litter. However, the impact of poultry litter application on heavy metal accumulations in the soil profile and the crop grains was largely unknown under Tennessee production conditions. So we did this study to look at the heavy metal contents in the entire soil profile and their concentrations in corn and soybean grains based on some long-term field experiments with and without poultry litter application. So this study was conducted on the UTIA Research and Education Centers at Milan in West Tennessee and at Spring Hill in Mid Tennessee. We used the two long-term field experiments relating to cropping systems and the biocovers, which were initiated in 2002. The two cropping systems we used for this study were continuous corn, means we grow corn every year, and the continuous soybean. And the poultry litter application levels were 4 tons per acre annually as a legend source, and no poultry litter application at the check. These are the approximate contents of nitrogen, phosphorus, potassium, and other nutrients in the poultry litter we used for the past 15 years. I mean, there were some changes, variations among years, but probably on average, these are the values for nutrients from poultry litter. Corn and soybean grain samples were collected from each plot at harvest in 2017. Soil samples were taken at 0 to 15, 15 to 30, 30 to 60, and 60 to 90 centimeters after harvest, also in 2017. Heavy metal contents in this soil and crop grain samples were determined. The heavy metals we tested included arsenic, cadmium, chromium, copper, lead, and zinc. The total content of arsenic, cadmium, chromium, or lead was not increased after long-term application of poultry litter. However, the contents of total copper and zinc was significantly increased after 15 years application of chicken litter. The total zinc content was increased by 1.8 and 1.6 times with poultry litter application relative to no application at Milan and Spring Hill respectively. Similarly, application of poultry litter increased the total zinc by 17% at Milan and 11% at Spring Hill. Poultry litter application increased the total copper content in surface soil 0 to 15 centimeters, as mentioned earlier. However, there was no increase in total copper in deep soil with poultry litter application. So the increase only occurred in the top layer of the soil. And similarly, applying poultry litter increased the total zinc content in the surface soil compared with no poultry. No impact was observed on zinc in deep soil. Long-term application of poultry contributed to the accumulation of available zinc in the top 15 centimeter layer was increased by 4.1 times a spring hair and 4.7 times a minor. However, poultry application showed no impact on available copper in deep soil. So available carbon showed the same trends as total copper. Similarly, long-term application of poultry increased the accumulation of available zinc in the surface soil. However, applying poultry had no impact on available zinc below the top layer of soil. In order to further characterize soil contamination by heavy metals due to poultry applications, 3 contamination indices, including contamination factor CF, degree of contamination DC, and the pollution loading index PLI, were used in this study. The CF is the heavy metal index. The sum of the contamination factors for all heavy metals denotes the contamination degree of the soil. PLI is used to estimate the severity of soil pollution due to poultry application. These are the classification criteria we used for the 3 indices in this study. Here you can say if the contamination factor is below 1, we say there's no contamination. If the DC is below 8, we say very low degree of contamination. If the PLI is below 1, we say there's no pollution. So based on those criteria as shown in the previous slide, generally no heavy metal contamination was found as spring hill in mid-Tennessee, because the CF values of heavy metals were mostly equal to or lower than 1, except for copper. However, moderate heavy metal contamination was presented at Minot in West Tennessee. Overall, the results indicated long-term application of poultry caused a moderate risk of heavy metal pollution in the surface soil in West Tennessee, and the copper probably had the highest potential for pollution, followed by zinc. So now let's look at the heavy metal concentrations in corn and soybean grains. These corn and soybean grains are used to feed animals or human beings. So here you can say application of poultry increased the zinc concentration in corn and soybean grains by 7 to 17% at both locations. However, the concentrations of the other heavy metals in crop grains were not increased with poultry application. So zinc was the only heavy metal with increase in crop grains. Whether that is good or bad, let's look at some facts. Approximately 25% of the global population is at risk of zinc deficiency. And zinc deficiency is characterized by slow growth, loss of appetite, impaired immune system. It is also a leading cause of infant mortality. So increase in zinc in corn and soybean grains is beneficial for animal health and for human health. Zinc Bio Fortification is a technique in which the zinc status of the edible portion of plants is improved by spreading zinc solution on crop canopy or through soil application. So it is encouraged to increase zinc concentration in crop grains due to the deficiency of zinc in humans. Therefore, poultry is a very good source to increase zinc concentrations in crop grains and then improve zinc nutrition for human beings. So these are the conclusions from this study. Long-term poultry application did not result in any increase in arsenic, cadmium, chromium, or lead in soil profile or crop grains under our production conditions. However, long-term poultry application significantly increased the total zinc and available zinc levels in soil surface and consistently enhanced the zinc concentration in corn and soybean grains, which is beneficial for animal and human health. Contents of total copper and available copper were enhanced in surface soil due to long-term application of poultry. The good thing is copper concentration in corn or soybean grains were not elevated. Overall, application of poultry at 4 tons per acre per year based on crop lighting requirement is generally safe, except some copper and zinc accumulation in the surface soil in Tennessee. Now let's move on to another topic, soil health and the carbon footprint on the major cropping systems in Tennessee. In this topic, we're going to look at the definition, indicators, and the analytical methods for soil health, the functions of soil microbes in soil health, long-term effects of cover crops, crop rotations, no-tillage, and their interactions on soil health, and the carbon footprint on the major cropping systems. So what is soil health? Soil health is referred to as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. So the key is that soil is a living system. Soil contains living organisms that perform functions required to produce food and fiber when they are provided with the basics for life, such as food, shelter, and water. Therefore, it is very important to maintain soil health so they are sustainable for the future generations. The Soil Health Institute in North Carolina recently proposed 19 soil health indicators, including organic carbon, pH, water stable aggregation, crop yield, soil texture, soil penetration, resistance, nutrients, etc. So you can say soil health has a lot of content. It is really a complicated concept. A healthy soil can provide the following functions, such as physical stability and support, nutrient cycling, water infiltration and availability, filtering and buffering, and the habitat for biodiversity. So how to keep a soil healthy or how to improve soil health? Here are some basic principles. Minimize soil disturbance, including mechanical, biological, and chemical disturbances. Keep soil covered all the times with plants or plant residues. Maximize diversity of plants in rotations or with cover crops in the winter. Keep living roots in soil as much as possible. Create the most favorable habitat for the soil food web. And if it's possible, integrate livestock into the farming system. This is a traditional soil testing report. Even now, we do soil testing in this way. Here you can see basically we only test some chemical properties of the soil, such as soil pH, soil organic matter, soil nutrients, including nitrogen, phosphorus, potassium, and then maybe even micronutrients. That's it. We do not test any physical or chemical property or biological properties of the soil. So we use these tests to make fertilizer recommendations. So that is our traditional soil testing method. So how to test soil health? It's much more difficult and much more complicated compared to our traditional soil testing method. Cornell University recently proposed a method for testing soil health. Here you can see it includes physical properties, biological properties, and chemical properties. For each category, it tests 4 parameters. So it's a very well balanced test in terms of different soil properties. So this method has been used in New York State and some other places. It costs over $200 per sample. And that may be too high for producers to accept. This is another soil health test method proposed by Rick Hannon at the USDA LRS. His method is different from the one proposed by Cornell University. Here you can see he only measures soil respiration, water extractable organic carbon, and nitrogen. Then a soil health score is calculated with this formula. And he also recommended to use methanium citrate plus 3 weak organic acids to extract the soil for available nutrients, then use the results to make fertilizer recommendations for the crops. So basically, this soil test method focused on soil biology. The Oxford company in Oregon used the soil food web analysis to test the soil health. This is a report from the company. You can see this method measures the biomass of different microorganisms such as bacterial, fungi, protozoa, nematodes. It also recommends, it also determines the plant available nitrogen supply. So this is a method also focused on soil biology. This graph shows the entire food web in the soil. From the left to the right are the first, second, third, fourth, and the fifth are higher trophic levels. Anyway, there are many organisms in the soil. Probably there are more than what we thought. Many different kinds, and they make up of the food web for the soil. Here are some very interesting numbers showing how much of each type of microorganisms in the soil. For example, there are about 10,000 pounds per acre of fungi, 6,000 pounds per acre of bacteria, about 2,000 of earthworms, and 400 of protozoa in the soil. Again, there are lots of organisms in the soil, and they are very important for soil health and crop production. So now let's look at the functions of microbes in soil health. The first function microbes in soil health is that they are the factories to make organic matter. The microbes can use plant residues, manures, or even their own residues to make soil organic matter. The active pool is called Nibble SOM. This stable portion is called the soil humus. So we need microbes to make soil organic matter. Is soil organic matter important? Surely it is. Here you can say this is a list of the functions soil organic matter has. Soil organic matter can increase water holding capacity, increase water infiltration, store nutrients, buffer temperature, provide appropriate soil structure, and provide homes for soil organisms. Another function of microbes is to make glues to stick soil particles together, which is beneficial for air and water movement in the soil. The third function of microbes is to make nitrogen fertilizers for plants to use. Basically, all the legume species can use nitrogen gas from the air to make ammonia for crops to use. In this way, it can save the use of synthetic nitrogen fertilizers. That is why we usually do not use any nitrogen fertilizer for our soybean crop. The soybean roots can fix nitrogen. And also, we recommend to reduce nitrogen application by 50 to 70 pounds per acre if there is a winter cover crop and that cover crop is legumes. The fourth function of microbes is to team up with crops to make crops healthier. These are two examples. Mycorrhizae. It is an association of fungi and the roots. Mycorrhizae can increase water and nutrient uptake by the crop, so it can make plant to be healthy, particularly under severe conditions. Another example is rhizobium. It is an association of roots and bacteria. As I mentioned earlier, rhizobium can convert nitrogen gas into ammonia for plants to use. Another function of microbes is that diversity communities of microbes can keep the bad organisms down. For instance, the vampiricides can eat some bad fungi, so can reduce some diseases. Anyway, microbes play a lot of roles in soil health and even plant health. So we need to care the organisms in the soil very well. as we do for our crops. These are the two basic rules to take care of soil organisms. We need to provide enough feed and water, enough food and water for organisms in the soil. We need to provide homes for them. If we do that, the soil organisms will provide nutrients for the plants, will build soil organic matter for good soil health, will aggregate soil particles together for good soil structure, and will help plants to resist stresses. Soil health research has been a hot topic during the recent years. So we did a project collaboratively at UTIA. This project was funded by the USDA Conservation Innovation Grants. 3 long-term trials at Jackson and Milan, and 6 private farms in West Tennessee were used for this study. We investigated the long-term impacts of cover crops, crop rotations, or we say crop sequences, conservation tillage, and their interactions on soil health, nutrient cycling, water availability, and the crop productivity. So what are the benefits with growing cover crops? This is a list. It can improve soil structure and fertility, reduce soil erosion, decrease weight, pest and disease incidences, lessen groundwater contamination, lower production contests, increase crop diversity, improve soil infiltration, and enhance soil organic matter. So there are various benefits with cover crops. Similarly, conservation tillage, particularly no tillage, provides a variety of benefits, such as reduce soil erosion, save labor, reduce energy use, improve soil health, improve water quality, etc. Compared with monoculture, crop rotations is good for improving soil health and productivity, distributing economic risks, and workloading, improving internal resource utilization, and reducing impacts of pests and the pathogens. So now let's look at a few slides about the results. This slide shows the effect of cover crops on sulfur nutrition of the following cash crop. Compared with no cover crop, here you can see heavy wheat and winter wheat as a cover crop significantly increased the leaf sulfur concentration of the following cash crop, cotton. This trend was consistently observed at different growth stages, early bloom, mid bloom, to late bloom. So our results suggest winter cover crop is a very good sulfur source. Winter cover crop can reduce or even fully replace the application of synthetic sulfur fertilizers to the following cash crop. So this will add some economic return for growing cover crops in the winter, mainly because the sulfur deposition from the atmosphere to the soil was substantially reduced due to the tightened environmental regulations. More and more sulfur deficiency problems have been observed on our crops. So cover crops are a very good source and a cheap source for sulfur nutrition for cash crops. Tillage effect, I always say no tillage effect on soil organic matter. As I mentioned earlier, soil organic matter, or I always say soil organic carbon, is very important for soil health. Here, we compare no tillage with conventional tillage in different soil depth in terms of soil organic carbon. You can say on average of the top 15 centimeter layer, there was no difference between the two tillage systems. However, in the very top layer of 1 inch, no tillage resulted in higher organic carbon content than conventional tillage. But at lower layers, let's say 5 to 7.5, 7.5 to 10 centimeters, no-till actually had lower organic carbon. So this result shows it is not easy for us to increase soil organic matter under Tennessee production conditions, even under no tillage. Recently, I had a PhD student who did some carbon footprint calculations for some major cropping systems. Here you can see we used a long-term study at Milan. The study was initiated in 2007. So we used the 15 years of data from 2002 to 2015 to calculate footprint. This is the CO2 equivalent emission from inputs per year for crop production. We had 6 different crop rotations: continuous corn, continuous cotton, continuous soybean, etc. Here you can see continuous corn had the highest emission from. Why? Because we use much more nitrogen fertilizer for corn production compared with cotton and soybean. Look at this figure. The CO2 emission from inputs, we have different fertilizers, we have herbicides, pesticides, fungicides, and the machinery use for planting, fertilizer application, harvest. You can say nitrogen contributes about 70% of the equivalent emission. So nitrogen is the major factor for emission from inputs. And on the right is a simple regression with the emission was fertilizer application. So it's very important to reduce nitrogen application if we want to reduce the emission from inputs. This is the output. That means the CO2 sequestered in the crop biomass. You can say continuous cone also had the highest output of CO2 equivalent emission, because corn produces much more biomass, including roots, above ground stalks, and grains. So net CO2 equivalent gain per year, this is one way to look at the footprint. Basically, it's the difference between output and input. Here you can say continuous corn had the highest net CO2 equivalent gain because it produces more biomass. This is another way to look at the footprint called the sustainability index. Basically, we use the difference between output and input, then divided by the input. In this way, the result is different. You can see continuous corn had the highest sustainability index. So these are the conclusions. Cover crop was a good sulfur source for the following cash crop. Long-term tillage systems and cover crops only impacted the top layer of soil chemical properties in West Tennessee. Nitrogen fertilization was the dominant factor, contributing to CO2 equivalent emission from inputs. Continuous corn had greater net CO2 equivalent gain than the other crop rotations. Continuous cutting resulted in higher sustainability index than the other crop rotations. At the end, I would like to thank the supporting staff, graduate students, undergraduate interns, postdocs, visiting scientists, collaborators, colleagues, UTA, RECs, and the funding agencies, including One Health Initiative, for their support. With that, I'm open to any questions you may have.
Thank you so much. What a wonderful and varied talk. So everyone, please, it's Zoom, but you know, I'm sure that there would be a lot of applause in person. I think maybe also either if you would like to type questions into the chat, I can read them out, or if people would like to unmute and ask questions, please feel free. I don't see any. Oh, here we go.
Yeah, I have a quick question for Dr. Ying. So you mentioned when you use the poultry litter as fertilizer, you see the copper and the zinc concentration kind of increased in the soil. So if you use, say, cow manure, do you expect a different set of metal will be increased? So do you expect differences between different animals in that sense? Yeah.
Okay, that's a very good question. And I mean, maybe not. It really depends, you know, on the feeding program for the animals. If they feed, you know, the feedings have like high concentrations of copper and zinc, then probably the performance will be similar as what we got from the chicken litter. But if the feeding formula is different, and then we may not see the accumulation of copper or zinc from other organic manures.
So the next question is, so those heavy metals, are they come from the feed or they are added to prove growth of those animals? So where were the ultimate source of those metal?
Okay, that's a good question. I'm not very familiar, with the animal feeds, but I guess probably mainly from the additives. It's, the fodder such as, forage corn, or like even like corn, soybean grains, are used as a feed for animals. They don't have very high concentrations of heavy metals. So my guess, probably the major portion is from the additives.
Thank you.
You're welcome.
Thank you. So we have another question or two in the chat. So if it's okay, I will read them out. So
Sure.
Awesome. So the first question in the chat is, what is the allowable limit of heavy metals in drinking water for humans? And which bacterial genera are dominant in healthy soil versus non-healthy soils?
Okay, I couldn't remember the the criteria or standards, for healthy water in terms of heavy metals, but I'm sure there are standards for bacteria, for fungi, for heavy metals, even for nitrate, for drinking water. But sorry, I'm not, I can't remember that, and so that is the answer. In terms of bacteria for soil health, I would say need to keep a balance of different kinds of organisms for the soil. We need a good balance between bacteria and fungi, between fungi and nematodes, between nematodes and protozoa. So in that way, the soil will be healthy in different aspects and will be a balanced ecosystem to perform many different functions. So I would say it's difficult to say which one is more important than the others. I would say all of them are very important in an appropriate range of their populations.
Excellent. Thank you so much. Yeah, I will ask the next question in a second, but I wanted to say I personally very much appreciate the perspective, not only from One Health, but just as in biology, of looking at sort of ecosystem diversity as a measure of health, specifically as a soil management perspective. I love that in your talk. Thank you.
You're welcome.
So our next question in the chat was, did your estimates of the CO2 emissions and corn biomass left in the field after harvest account for the relatively rapid degradation of much of that biomass over time?
Okay, so when we calculate the footprint, basically, we is assured we use the inputs, all the chemicals, machinery use in terms of CO2 equivalent emission. So that's the input. For output, we use the whole crop, the sequestration in the whole crop biomass, including underground roots, above ground leaves, stems, and the grain yields. However, we did not consider the soil organic matter in the soil. So our box only covered the whole crop at the output, and all the chemicals at the input.
Thank you. So there's a follow-up that is related a little bit already to what you have answered, but I think there's still more. So did your estimates also of CO2 emissions from nitrogen fertilization include energy required to manufacture the nitrogen fertilizer in Haber-Bosch process? forgive me, I don't know what that is. So
Yes.
presumably that's the
Yes,
question.
let's say for nitrogen fertilizer, we included, the production of nitrogen fertilizer in the nitrogen company, how much energy was used. We also included the transportation of the nitrogen fertilizer from the company to private farms. And we also increased the machinery use for nitrogen fertilizer applications. Yes, so we increased those into the calculation of nitrogen fertilizer. And similarly for other chemicals, let's say fungicides, we included the energy used for manufacturing that fungicides and the transportation, the energy for transportation for that chemical to be on the private farm.
Wonderful, thank you. So I'm cognizant of the fact that we've exhausted the hour, and I know people might have to go, but hopefully if there are more questions, people will feel free to reach out to you. Thank you. Let's everybody thank Dr. Yin again for such a wonderful talk and for such excellent insights from the question and answer. Thank you so much. What a great way to close out our seminar series for the semester. Everyone have a wonderful December, and we will see you again in January. Dr. Yin, thank you again. One health, wonderful things to everybody.
Thank you all.
Thank you.
Thank you. Goodbye.